Alright guys, let's get the first chapter started here. We're going to talk about muscles. In my mind, swimming or any athletic sport really is all about how you move the body, right? And the only cells in the whole body that do produce any movement are muscles. So that's where we're going to start. And you'll notice the first, part one of these courses is all about how muscles work, how they're controlled, how they get energy, how they, are controlled from the nervous system. So basically everything from here on out is going to build on a good understanding of how muscle works, but we first have to understand how it works. In my mind, I always like to start with where we're trying to go, what's our destination, and try to be as specific as possible. So with muscles, the whole goal of a muscle is to get shorter. That's really the only thing it can do. And the reason it wants to get shorter, it brings two bones closer together, right? So that's how you close a joint. So how does it do that, and what are some distinguishing things that make that possible or make that different between different muscles? That's what we're going to start with, talking about sarcomeres, myosin heads, and then the second part of this talk is we'll try to relate it back to the pool deck and see if we can't take some of what we learned about how muscles work and apply it to what we see and what we feel and what we know about swimming. So just like I said, muscles bring bones together, and the way they do that is with something called a sarcomere. So this is kind of our base unit. If you zoomed all the way in onto a muscle cell, you would find these repeating units called sarcomeres. And this is actually what shortens and what gets smaller. And if you line up a whole bunch of sarcomeres together, millions and millions of these in a single muscle cell, you'll get an additive effect, right? So this, you might be able to see here, like this amount of distance is a little bit shorter than that amount of distance when we shortened up. So it only might be nanometers for each sarcomere, but it's going to be millimeters, inches longer if you add it up over the whole length of a muscle. So you have millions and millions of these inside each muscle cell. We're going to zoom in a little more and go into it in a little more detail, but here you can kind of see the additive shortening part. So if you line them up, and then this picture down here is an actual electron microscope picture of a real muscle cell. So you'll notice that there's these things called the Z line here, these black lines. So these kind of act as anchors, and then the red lines, and actually we'll go into it more detail. Red lines, if you've ever heard of thin filaments or actin, and then you have these blue lines, which are thick filaments, and myosin, and you can see they kind of relate to what we see right there. And in the areas where it overlaps is this kind of thick dark band there. So just real quick. And you can see here the bottom picture is where you actually have a contracted muscle versus a relaxed muscle, and you see how the Z lines got closer together. So when sarcomeres work, when they turn on, they try to bring those Z lines together. So let's figure out how that happens. So here I've split up my sarcomere